A university campus. 3,000 students walking between classes, labs, and dorm rooms. The career counsellors question, which of these students will become doctors? Which will become engineers? Which will end up in research? She can't follow each one individually, but she can survey them, map them, and predict their trajectories. This is the multi-omics to fate pipeline, and every step has a biological counterpart. The counsellor collects 3 types of data on every student. Academics, scVI, course selection, test scores. This is transcriptomics, measuring what genes are actively being expressed. Extracurriculars, clubs, volunteer work, sports. This is epigenomics, measuring chromatin accessibility and regulatory activity. Personality assessments, value surveys, career interest inventories. This is proteomics, measuring surface markers and protein expression. Each data type captures a different facet of the student. MOFA Plus, multi-omics factor analysis, integrates all 3 into shared latent factors that capture the common variation across all data types. Not all factors matter for career prediction. Random Forest train on the MOFA Plus factors reveals Factor 1, driven by biology GEA and hospital volunteering, has importance 0.85 for predicting medical school. Factor 3, driven by coding courses and hackathon participation, has importance 0.78 for engineering. Factor 7, driven by art portfolio scores, has importance 0.02 for the medical path. It's irrelevant. The counsellor throws out factors with low importance and focuses her advising resources on the factors that actually predict career outcomes. Using the important factors, the counsellor builds a map of every student's position relative to every other student. But there's a problem: different schools grade differently. A 4.0 from a grade-inflated school isn't the same as a 4.0 from a rigorous one. The single-cell variational inference model accounts for these batch effects. School identity goes into the decoder, but not the encoder. The latent map reflects student ability, not school grading policy. The batch-corrected map is the honest view, where students truly sit relative to each other. The map reveals structure. Clusters: pre-med students cluster together in the upper left, high biology DPAS, hospital volunteering, MCAT prep. Engineering students cluster in the lower right, coding courses, robotics club, linear algebra scores. Liberal arts students form a diffuse cloud in the centre. Trajectories: undecided freshmen sit in the middle. As they progress, they stream toward their eventual cluster. Pre-meds drift upper left, engineers drift lower right. The map is a UMUP projection of the scVI latent space. Students with similar multi-omics profiles sit near each other. The biological translation is direct. Students are cells. Academic performance is gene expression, transcriptomics. Extracurricular activity is chromatin accessibility, at-seq, epigenomics. Personality assessments are surface protein expression, site-seq, proteomics. MOFA Plus integrates the multi-omics layers into shared biological factors. Random Forest identifies which factors predict disease state or cell fate. scVI produces a batch-corrected embedding or cells from different experimental batches sit in a common latent space. The map is the foundation for everything downstream. Survey across domains, multi-omics integration with MOFA Plus. Find what matters, feature importance with Random Forest. Map the students, batch-corrected embedding with scVI. The counsellor now has a clean, informative map of every student's position. She knows where they are and what predicts where they'll go. Next, predicting the destination.